So I'd like to welcome you all to the Cancer Center seminar series. It's a real pleasure to introduce, he's got his back to you right now, but Mahmoud Khalifa. Mahmoud is the director of, or the division leader of anatomic pathology in the pathology department. And he, just a little bit of background, he got his medical degree from Cairo, Egypt, and subsequently received his master's degree and PhD in pathology. I don't have any of those. Continued his training at University of Oklahoma, George Washington University, Georgetown, and the Armed Forces Institute of Pathology. So he is extremely well versed in pathology, in anatomic pathology, and he has had just an amazing career. He joined us when? 2 years ago?
A year and a half.
Year and a half. I rounded up. And so what I want to emphasize is when he came to us, he fundamentally redesigned how the pathology department is put together and designed around a model of subspecialty. It does two things. The faculty members who are now in pathology have the latitude to be able to interact with people doing research because they'll have more time. And they also have a subspecialty so that you can consult with people who actually are a specialist in a particular disease. You know, we have a lot of omics coming out these days. There's no shortage of omics, but they need tissues in order to validate them. And pathologists need to be fundamentally involved upfront in those kinds of studies. And so Mahmoud is going to talk to us today about some of the things that he's put together in the department so that people can access them. And I urge you all to do so. Mahmoud, thank you very much.
Thank you for the introduction, Jim. You summarized basically my talk. The whole subject of tissue-based research is very complex, and I just want to state upfront that when I came here and saw how complex our system was, I decided to dissect the issue. There's a lot of noise, a lot of strong emotions about the whole thing, and I am dissecting it, and what we are going to talk about today is a very specific segment of that whole spectrum of tissue-based research, which is the access to archival material for research. I have nothing to disclose, and I will be using some cases in this presentation that are inspired by real cases, but they are not the real patients. So what I am going to be doing today is to go over the current state of affairs, discuss with you some of the things that you already know, but from maybe our side of things. And I'm going to share with you some stories, and the objective is to bring you into our world because I realize that sometimes researchers and clinicians speak different languages. And hopefully this will start a dialogue about creating an intelligent organization-wide solution that can help us all in what we are trying to do. And I will take this opportunity to present what I consider as phenomenal work done by our colleagues, members of the team here, and I will introduce them to you in due time. So when we talk about tissue-based research, I'm just trying to classify things to make things a little bit more meaningful. Let us assume that tissue-based research can come in two flavors, prospective and retrospective. And by that, I mean prospective research is when a specimen is solely removed for research purposes and it is handled fresh. And in that scenario, you already know what the specimen is, and you are not expecting pathologists to give you any diagnosis. If this is the kind of research you are involved in, then you need to talk to Bionet. And I want to separate Bionet from anatomic pathology from the get-go here, because Bionet's mandate is very different from pathology. Bionet is an organization in a broad sense that is meant to help you obtain this fresh tissue for research when no pathology services are needed. On the other hand, if you conduct a retrospective research, you are tapping into archival diagnostic material, and that usually is not fresh. That is formalin-fixed, paraffin-embedded tissue, And in these cases, they already have a diagnosis, or you need a pathologist to make a diagnosis, and this is where you should contact anatomic pathology. So this is one way of looking at the big picture. Maybe another way of looking at it, well, we can look at research as clinical trials and non-clinical trials. And this is a big thing because the rules applied to clinical trials are very different. Non-clinical trial diagnostic tissue is part of patient's permanent record, and it is our mandate to maintain that tissue to remain compliant with the College of American Pathology guidelines. As an accredited anatomic pathology lab, and now I'm talking about Fairview, Fairview is required by law to maintain permanent patient records. So we are not allowed to deplete tissue from our bank just because of research. You can come back and say, well, but maybe we can if it is clinical trial. And I strongly support that. That actually is one of my very strong points of view. But then how can we draw the line? What is a clinical trial and what is non-trial research, retrospective research? For the purpose of this presentation, and maybe we can work on that definition, I see clinical trial is a study that a particular patient is going to be affected or benefited. Non-clinical trials are about a series of patients, and you are not aiming to help one particular patient. That is a very loose definition, but at least it is a definition that allows us to draw that line as to when we can deplete the tissue from the bank. So it's about a year ago that we all saw this memo from the Dean's office emphasizing the need that any tissue removed from the OR needs to be accessioned in anatomic pathology, irrespective of the purpose. And again, you can see here the separation between clinical care and research. If it is for research, a specimen removed for research, clinical trial or non-clinical trial, it is fresh, it goes to pathology, it gets a number in our system, which is called COPATH, and you will hear COPATH a lot today. So it gets a number in COPATH, but we accession a number called MT, which is technical work, which does not require a pathologist to release a report. The case flows through anatomic pathology, gets a number for tracking purposes, handed over to Bionet, and then handed from Bionet to the researcher, if the case is purely for research. On the other hand, cases for clinical care, they are accessioned under, in anatomic pathology, they are assigned a U or an M number, and that's an internal number for us that trigger a sequence of events, ends up by a pathologist releasing a report on that case. That's very simple. We have seen this memo a year ago, and we have been trying to implement that policy. Of course, everything gets complicated because we realized that the policy was good, but it did not answer some questions. It did not say what or where or who. And by that, I mean, what kind of tissue are we talking about here? Are we talking, what kind of specimen? The memo talks about specimens. Is it a tissue specimen? If it is tissue, it's anatomic pathology. Or is it body fluids? Or is it something else? So that needed some tweaking down the road. Where is the specimen removed at? The memo talks about OR. So what about endoscopy suites and what about IR and that sort of thing? And who is it going to be in charge of implementing this policy? So yes, there is a policy, and yes, we are trying to tweak it and make it more applicable. About 4 or 6 months of me into this job, I had my first encounter where a researcher walked into my office, a very distinguished researcher from this organization, walked to in my office, was obviously not very happy, was not the happiest researcher. And the reason he was unhappy was because Fairview is blocking his work. Fairview is not releasing the slides that he requested. And I asked him what was the study about. He said this was a liver multi-center study. I require the original clinical slides and clinical blocks to be sent centrally to another lab. It's multi-institutional, and it is reviewed, and additional sections are obtained from the block, and we conduct our research. We have been doing this for years. What's wrong with you? So I asked him, well, so who is the pathologist with you on this study? He said, no, you're not hearing me. I don't need your pathologist. We have a central pathologist who is reviewing these cases. I said, yes, but our pathologist needs to ensure the quality of the material we are releasing to you. And our pathologist is needed to ensure that we maintain patient's record. He didn't seem to understand very well what I was talking about. And for those of you who have not seen clinical material or paraffin block, this is how the paraffin block looks like. Some of us here can look at this paraffin block and say, well, there is plenty of tissue remaining in that block. You can cut probably 50 or 100 sections, and I probably agree with that. But if you look at this other block, maybe one or two people just in this room who can see that there is a little bit of tissue here, and that's all what this block has. This little bit of tissue maybe can give you 2 or 3 more slides, and that will be it. So which block are we talking about here? Because, again, we are not supposed to deplete patients' record. And then what about the cost involved here? Because when Fairview retrieves blocks from the archives and cuts sections, there is cost. And I don't know what the slide costs, but there is. He said, no, you can't do that now. You cannot talk about money because we did not budget this from the very beginning. You can't come in the middle of the game and say, now you are going to charge me $10 a slide, and I don't have money for this. This is too much. This is very obstructive. And I could clearly see that I was having a problem. I am working in an environment that is very different from what I was used to. There was a problem because there was no pathology endorsement, and by pathology endorsement, we need to verify that there is an IRB approval. We need to verify the quality of the sections we are releasing, and we need to make sure that that we are compliant, and there was no oversight to ensure that compliance of maintaining patient's record. And there was no reimbursement mechanism, and there was somehow a pro bono culture in pathology. So pathology was supposed to be just releasing tissue and catering to every researcher's demand without being compensated. And to be honest with you, I looked internally in the lab, and there was not really a very clear process in our lab also to ensure timely delivery of these specimens. Let us dive a little bit more into what I'm talking about here, and let us think of yourself now. Pretend that you are on a review board for this study, and here is a researcher that is applying for this project. Let us read the proposal and see if you agree with this proposal. So this researcher is interested in somatic gene mutation in ovarian serous carcinomas, and they wanted to focus on BRAF. And in their study, they want to study V600E testing in BRAF gene. It will be performed on fresh tumor tissue. So the patient will be consented. We will take a sample from her tissue to submit it for molecular testing. But they also wanted to correlate the rate of mutation between pre- and post-operative CA125 in blood, which is obviously a very relevant question. But acknowledging that some of these patients may have received new adjuvant therapy, so at time of surgery, you will not be able to see residual tissue. For these patients, they will wait until it is processed in pathology, and the pathologist decides which particular block has any cells at all, and they are going to use that cell for their research. What do you think of this proposal? And let us imagine that this proposal goes to IRB for approval. I think it's a very good, it's a very solid project, and it flows very well. It flows very well in your head now, and it flows very well in my head. And this reminds me, if I ask you, how do you drive to Chicago from Twin Cities? Well, I get on I-94 and I just hit east and I will get to Chicago. It flows very well. Here's a highway that goes all the way from Twin Cities to Chicago. Well, that probably is true, but if you try to do that, and I've tried it many, many times, you will realize that after about 40 minutes of your drive, the speed limit changes. As soon as you cross Wisconsin state line, the speed limit becomes 70. In Minnesota, it is 65, and it stays 70 to Chicago. Okay, and then you realize that as soon as you cross Illinois state line, you will have to pay money now because it's a toll road. You didn't pay money in Wisconsin or in Minnesota. So the point here is that we do that every day. Things make sense in our head, and when we start implementing multi-step project, we realize that we are crossing multiple lines, and what we do is to comply with the rules and regulations in every place. So the idea here is that this researcher should be mentally prepared that while the research is very logical, they are crossing multiple lines here. Because the first line, if you are looking at fresh tissue, that is fresh tissue for research, and that you go to biobank and you get that from them. If you are correlating with body fluid follows a whole different set of regulations. But as soon as you want tissue, as you claim that you want tissue from pathology, that takes you yet into a whole different ball game, and we need to be aware of that. So if you are going to take anything away from this talk today, it will be this table, which clarifies 3 pathways for 3 distinct subtypes of specimens that were in the memo that we saw. Again, fresh tissue, you register with Bionet, and you take that from the OR or the other location where the tissue is harvested. It goes to surgical pathology, pass through just to be documented for tracking purposes, goes to Bionet, and from Bionet to your hands. Body fluid is a whole different story because body fluids goes directly from the patient to the researcher's hand. Archival tissue is a different story. You need to register with anatomic pathology because you are tapping into AP archival tissue, and you get a pathologist to work with you and will be accountable to me and to the organization to maintain patient's record, and the tissue will be handed to the researcher. So 3 distinct subtypes because these are 3 distinct state lines. This is just a slide to highlight the differences between these 3 different types of specimens. Now, come closer to our world, and I want to share with you the story of our dear Mrs. Block. This is what we do every day. This is my bread and butter every day. So Mrs. Block in 2010 was a 72-year-old with postmenopausal bleeding. She was experiencing fatigue, loss of weight, and increased abdominal girth. Probably she was having ascites. She went to her family doctor, referred her to some imaging testing. She had a CT, and she was found to have ascites and an omental cake. She was referred to IR, who stuck a needle, a CT-guided biopsy in the omental cake to get tissue diagnosis of Mrs. Block's problem. But since Mrs. Block was very nice and was very pleasant, she was approached in her course by a researcher who was having an ongoing research and consented her, said, Mrs. Block, you are now going to have a specimen. This is cancer. We have this project going on. We will need access to your material, your fresh material, to get some tissue and use it in our block. And she said, well, yeah, that's fine. And then she signed the consent. So this is the specimen that was removed. For some of us in the room here, we can clearly see we have like 4 different tissue fragments. I can tell you that each of these fragments is no more than maybe 1 or 2 millimeters in greatest dimension. These 2 fragments don't count because they are just blood and necrotic tissue. This fragment is probably necrotic, but we have some cells here, and this is where the money is, and this is where her cancer is. So with these specimens, as I said, this one piece of tissue, closer look here, you can see lots of necrosis, some inflammatory cells, and maybe some cancer cells down here. And for this specimen here, you can see that this is the material I'm working with. So this is the challenge I see every day, maybe 20 to 30 times. I am presented with such a small fragment of tissue with few cells, and I am supposed to deliver the highest level of service and provide the oncologist with as many information as they need to help this patient. We can all see cancer here. Probably it is high grade. Where is it coming from? I need to do additional stains. So I do immunostains. I do something like PAX8. You can see nuclear staining here, which confirms that this is Mullerian coming from the female genital tract. So now we are limited to maybe endometrial or maybe ovarian. How am I going to decide that? I do another stain like WT1. WT1 is positive in ovarian cancer, but negative in endometrial cancer. You can see it is negative here. And I do ER-PR to help the oncologist with their hormone manipulation further on. So with this panel, and this is a very, very basic panel, the idea here is that I started off with a very limited amount of tissue, and I consumed probably all of it in order to come up with the information I'm about to deliver to my oncologist now. So the diagnosis of metastatic high-grade carcinoma, probably serous carcinoma, from the endometrium was made in this particular case. Mrs. Block was too sick to have surgery, and therefore the oncology team decided just to treat her by chemotherapy. And by that, you realize that once the patient goes to chemo or palliative chemo, then there will be no more tissue taken. So all the tissue we have on Mrs. Block was those small fragments I showed you in the previous screen. As she consented for the research, 20 unstained sections were prepared and were shipped for the researcher, and that was gone. The patient did well, and 2 years later, she seemed to come back with a recurrence. And right at that time, there was a new clinical trial just opened and required molecular testing from the original tumor to ensure candidacy of the patient, because they were going to put her on some new intraperitoneal drug. They went back to the block to obtain some tissue, but the block was depleted. Because it was mostly necrotic. I used most of it to render the clinical diagnosis, and then the researcher used 20 other sections to participate in their research 2 years ago. Today, the tissue has been depleted. She could not come back for another procedure. We could not get back more tissue, and therefore the patient was declined from her clinical trial, and Mrs. Block passed away soon after that. And I'm not saying that Mrs. Block died because she didn't have a chance to be enrolled in this clinical trial. Nobody knows that. I'm saying that she died without being enrolled in that trial, and she was eligible for it, and she should have been on that trial. And as a director of anatomic pathology, it was my responsibility to ensure that the tissue still had, the block had some tissue to allow her for additional testing down the road. So the message I want to deliver today is that neither patient's consent nor IRB approval ensure full compliance. So many researchers come to my office and say, what are you talking about? I have an IRB approval. I say, yes, with all due respect, IRB looks into ethics issues from the patient's point of view and whether there is patient harm or not. But it doesn't tell you whether this particular patient, maybe you are enrolling 20 patients in your study, and maybe 19 of them have enough tissue in their blocks, but the one patient that doesn't have enough tissue in the block, it is my responsibility to ensure that we don't deplete that block. And therefore, neither IRB approval nor patient's consent guarantee that you are going to have access to clinical material. Access to clinical material will be rendered judging the tissue on case-by-case basis. So going back to the problem we had, we realized that I was having a problem, and I realized also that I cannot solve this problem on my own. I needed a lot of help. I needed help from a wide range of experts. Some of them are already in this room today. We pulled out our toolbox and we decided to work together to come up with a solution. And as a guiding principle, we agreed that our solution has to have these features. Our solution has to ensure high quality of the submitted material. It needs to preserve patient's permanent record and not deplete the patient's blocks. It needs to be efficient. It needs to be responsive to researchers' demands and needs, and it needs to be fiscally responsible to compensate our lab, and it needs to be sustainable. So with these guiding principles, I reached out for some leaders in the organization, and the first step they did was to look around, and they found this tool that already exists, Encore, as part of the CTSI mandate. And we probably are all familiar with Encore. And this is a system that is already in place, and maybe we can leverage that to help us find a solution to our issue. Many of you have seen this slide before. I borrowed it from the Encore demo about the pre-award overview and how during the pre-award overview, Encore generates a calendar, and pathology now wanted to jump on that calendar to try to find a solution. So here is the workflow that we developed, and it is still in the draft form. It is very, what's the word? It's not complex, but probably the print is too small for you to see. The idea about this screen here, it shows you that it goes into two phases, the pre-award and the post-award. And it involves three teams working in tandem. Here is the research team, and this is pathology, and here is the Fairview Research Administration. And each one will be following certain tasks according to this flow chart. Let us focus on the pre-award phase here, and it starts from the first step when the researcher decides that their proposed research is tissue-based. So I hope that we get into this habit that as soon as you declare your research as tissue-based, you will realize that this is something that we need to involve pathology in, and the mechanism for that is you will answer a question in Encore, and the yellow line, the yellow boxes here indicate that this is something that's still being developed. And we are working with Encore team to have this question for you: is it tissue-based or not? And if it is tissue-based, are you seeking fresh tissue for research only, or are you tapping into clinical archival material, or maybe you are trying to do both? Once that box is checked, an email will be sent by the system to pathology, and pathology needed to create a generic account that will receive these emails. And pathology also is working on creating a new position, which has been created already, and it will be posted this week or next week, for a research coordinator in pathology who will be receiving this email. Once the email comes to pathology, a study pathologist will be assigned. So now you will be assigned a pathologist, and that pathologist is going to contact you to discuss the proposal with you and go into details of your ask in pathology and whether this ask makes sense or doesn't make sense. And once there is an agreement with the pathologist, we will need to sign an agreement with the Department of Pathology. And I will show you a draft of this agreement now, and this has also been developed. Once there is an agreement with pathology, this agreement will be uploaded in CoPath, and it will be available for you to use it later on. The system is going to notify Fairview Research Administration, who is going to check your IRB approval and is going to create an account for you. So as you can see, this part of the process through Encore involved anatomic pathology and Fairview Research Administration. These are some of the roles that your study pathologist is going to work with you on. Will ensure that the tissue is not depleted, will ensure that because some of the, and I think some of the prostate projects that we are conducting now require processing of tissue in ways that are not necessarily the standard of care, and we are open to that. But who is going to ensure that every prostatectomy in that project is going to flow in this path in the lab rather than the routine? It is going to be your study pathologist. So the study pathologist ensures that your specimen will be handled according to the agreement that you signed with them. And we'll work with Fairview, if it is a retrospective study, to review the slides, select the proper slide, and select the proper block for you. And depending on the amount of work involved and the role of the pathologist in the study, if it is significant enough that warrants academic acknowledgement, the expectation is for the pathologist to be acknowledged in the authorship. If it is a multi-institution study, if there is minimal work involved in pathology, then there will be a professional invoice. There will be an invoice for the professional services rendered. This is a chronic issue if involving pathologists in study. And our colleagues in the University of Calgary, Jim Wright was the previous chair, and Martin Trotter is the current chair. They did a very interesting study. They went to the Canadian Journal of Medicine and screened all articles, multidisciplinary articles that have pathology images in the article, and found out that 47 of these articles don't even have a name of a pathologist as an author. So this means that a clinical team goes to pathology, ask a resident or ask a pathologist out of the goodness of their heart, I need a picture of that particular tumor, and they take a photo, they give it to them, and it appears in the article without any acknowledgement of pathology. And they took that as an evidence of under-recognition of pathology. Frankly, when we read that article, I mean, these are our colleagues, and we, of course, suffer from that chronic problem, but we felt that we needed to respond to their article, because the role of pathologists is not just about taking the picture. The pathologist generates the data, annotates the tumor for you, and works with you on collecting data, collecting information, and ensuring the solid pathology in your article. So it is not just 47% of the articles that are not recognizing pathologists. The problem is a lot more than that, and it's actually underestimated, because clinicians take informations and researchers take informations from EPIC and collate them and publish them. So here's an example of the draft. Here's the draft of the pathology agreement. And you can see that the agreement basically talks about 4 different services that you will be asking from pathology. Are you just collecting data, or are you asking for technical work? And here's some examples of the technical work you will be asking, and you may add some details here. If you are asking for professional services, a pathologist, what level of services will you be asking? Is it just a pathologist that will do on-site assessment for you? Some of the studies need a pathologist to be there at the time of acquiring the specimen. Or are you asking a pathologist to select the slide for you? Or are you asking for a full report on the case? Some other services that are being requested frequently from us are regarding shipping and handling of these specimens. So this will be the agreement that you will fill up at the time of application for the study. It will be agreed upon with your study pathologist, and it will be uploaded on Encore, waiting for the study to start. When the study starts, and that would be the post-award phase, by then you will have an IRB approval. We will have an agreement on site uploaded, and you will have an account with the Fairview Research Administration, so you are ready to go. When you feel you are ready to go, then you need to fill a form requesting the material from pathology. And this form will be emailed again to our research coordinator or research liaison in pathology. And that person is going to look at your request and go online to review the agreement that we signed maybe a few months or a few years prior at the time of studying the protocol, and reconcile the two and make sure that what you are asking today matches what was agreed upon previous. If it does not match, then they will contact the study pathologist, and the pathologist within our department will be contacting you to say, well, remember we agreed on this and now you are asking for this, and then you will say the reasons for that, and maybe we can change the agreement accordingly. Until we reach an agreement again, or maybe the request is matching the agreement, it will be handed over to the technologist on the bench, and they will cut the sections and prepare the material, whatever it is for you, and will hand it over to you. And this will trigger the billing part, which will be handled by Fairview Research Administration or by UMP, depending on whether you're asking for technical work only or professional work only, or both. So we feel that this process here streamlines the process a little bit and keeps everything on record, online. Everybody can go back and check, and it is transparent. And we revised the pathology request form that you will be using to request your material from the lab. And here again, it talks about technical and it talks about professional, and you will list the number of the cases that you are asking, and you will list the specifics of the sections, the thickness, whatever you're asking from the given block. So we also realize that nothing is simple in our life and things get very complicated. So even though we see research in these terms, I can probably say, and probably you would agree with me, that almost no two studies are the same. Every study has its own nuances and has its own specifics. So now we are running into studies that are prospective, and they are supposed to be, and because it is prospective, it involves fresh tissue, so the researcher contacted Bionet, and Bionet is involved. But guess what? Now the researcher also wants pathology to give them a diagnosis. Well, we already said that Bionet doesn't do that, but pathology does. Well, maybe we should combine the two, and now we are prepared to deliver this kind of service. And this was a clinical trial, and by definition, we will allow depletion of the tissue. But in this particular study, we needed a lot of work from the pathologist. So again, here's the flexibility that the new system is offering to you. We should be flexible. We should allow for all the permutations. And if there are pathology services needed, there will be agreement on the fee schedule, and Karen was just telling me that she is working on the fee schedule for research, and you will be presented with these upfront, so we don't run into that situation where we ask for reimbursement down the road. From the pathology point of view, this is how we see the process. There is a request phase, there is an approval phase, and then when the study is awarded, there is a technical phase, and then we have to when we release the tissue, you have to remember that every time we release tissue to you, we have to refile it back. So we recognize the tissue internally in the lab in these 5 phases for our personal management in the lab. So some of the work that is still in progress, we are very close to finalizing the agreement and finalizing the form. And once that is finalized, you will be notified and it will be available to you. We are making some adjustments on Encore because, as I said, Encore needs to ask you that question upfront, whether it is tissue-based or not. And as Encore creates the calendar, it will involve pathology steps in it, so pathology will be notified of the various components of your calendar. We need to finalize this flow map, and we need to update our websites. And when I say our website, and I mean Bionet and anatomic pathology, because if you go to the Bionet website today or anatomic pathology, God help you, it is very complicated. It is not matching what we are talking about here. It is not up to date, and pathology definitely needs to be more clear. And I was waiting on finalizing this process. Once it is finalized, once I get feedback from some of the stakeholders, we will be able to update both our websites in pathology and in Bionet. At the end, I just want to acknowledge members of the team that help us get this far, from the Fairview Research Administration, Jill, Julie, Krista, and Michelle. From Fairview Pathology, Jennifer Morocho is the supervisor of technologists, and Sammy is the supervisor of our office. From the UMP side and ALRT, Linda, Kathleen, and Karen here, our accountant. As you can see, the process involves so many aspects, and it is so diverse, and it needed the input from various people. And I need your input as well as to how do you see this flow. And I'm walking into this with the assumption that every study is registered with on OnCore. Of course, if you are not using OnCore, what is going to happen is, and this part here was designed in this particular way specifically for people who are not using OnCore or for external researcher. In other words, someone can shoot in from outside and just says, I want 20 slides from whatever. And the same process will start here. We will say, okay, then we need to see the IRB approval. You need to have an account, and you need to fill in this form to clarify the request from pathology. So it is, I think the process is so fluid, and it is versatile, and it allows for the various permutations in research. Thank you very much for being here, and I can take any questions.
That was great for putting that together, and I think having a structure on tissue access is incredibly important for us, because I think, as you pointed out, we really didn't have that. I do have a question about an issue you brought up in terms of how long do you have to keep the material legally. And the reason I bring it up is I was at a presentation recently from one of our colleagues at the University of Michigan. They ended up, as they were going to throw away blocks after the legal time it was required, they actually ended up creating a tissue microarray of everything they were going to throw out, and then linked it to a clinical record and made that available to the investigators. So I thought that was actually, and that some material was 10 plus years old, which is, for some people, it's very important to have specimens there that kind of duration, because the follow-up will be known. Of course, that leads to a whole other question about how do you link to clinical outcomes. But nonetheless, when you said you didn't want to delete the blocks, but at some point you're not going to keep them anymore. And I wonder if there's any reach-through, a way to keep those tissues for research purposes.
Excellent questions, of course. Maybe, Chris, you can answer us about the question about the requirement. How long are we required to keep the tissue? So 10 years. What happened, our system here, after 10 years, when Fairview doesn't have to keep the tissue any longer, the tissue is transferred to us here in UMP, and we keep the blocks here. Scott? Yeah. Yeah, so the older blocks, older than 10 years, are kept with us here on campus. There is a process involved, because we don't want to keep, we follow a certain set of criteria, for example, in oncology, and certain types of blocks are kept, but the others are disposed of. So the answer to your question is we keep our blocks for the value in research and academics indefinitely here on campus here. But you brought up another issue about tissue microarray, and maybe this is something that we need to talk about also down the road. As a group of researchers, maybe this is something that we need to invest in. For example, in Bionet, they have in their lab a very skillful technologist, and they have the facility to create PMAs. So maybe at one point we can come together and say, you know what, we need to create TMAs from everything. Well, in pathology, of course, because that's our bread and butter, we already have these projects going on. The prostate team is doing it. The ovarian team are doing it. So we are creating tissue microarrays for archival material. But maybe at an organizational level, we need to look into developing this down the road.
I have a quick comment on that. I mean, there's also higher throughput, newer technologies for TMA creation that if AHC wanted to invest in it, would make it easier for, So there are newer, higher throughput technologies for TMA creation. If AHC or a medical school, something large that has a big budget that could actually provide that type of instrumentation could help us as pathologists do that type of work much more easily than it would take right now to do that. Something that I've looked at with Doug actually for our breast project as well, and it does take a lot of time for the pathologist right now, which is a huge time resource, money resource. So.
Is there a database that the researchers can look at that describes what tissues are available within the archive? Because then that gives us the information that we need to start thinking about to follow these steps in order to accomplish the goals of our study. But if we don't know the N, how many tissues are available, or what kinds of tissues, the spectrum of tissues that are available, it's really hard to understand how we want to design our study and bring pathology into the picture.
So I came from an environment that had electronic medical record as EPIC running in one level, and you had CDR running at a different level, clinical data repository. The clinical data repository is a system that many universities in the country have it now, where researchers can log in and see almost the exact same EPR, but without patient's information. So you have access to all the clinical and pathologic information without patient record. For our purposes here, again, this is very simple, and it brings, again, the issue of teamwork and the study pathologist. Jim mentioned earlier our subspecialty model in pathology. Now we work in teams, and in your prostate or then GYN or all of these teams, you have members in pathology, and you can just ask the question. You can just ask. Actually, this morning I was, and I don't want to expand beyond this topic, but because we are looking now into adding a new system to pathology, which allows us for discrete data capture. And the example I gave it as a challenge to the team, I said, I need a list of all renal cell carcinomas, chromophobe cell type, between 2 and 4 centimeters from male patients with positive margins less than 30 years. And in 5 minutes, they responded, and they gave me 2 patients who actually fulfilled that criteria. That was in a system where they capture information in discrete data field. We don't have that here. If you want to design a study like this here, it will probably take forever just to find these patients. So, but that is also happening and coming in the pipeline to be able to capture data as discrete element like that. But from the pathology point of view, if you work with a pathologist, we still do the natural language search to find you what you want. And that's the best we can offer now.
So since I actually have a number of the important people in the back that are kind of can answer this question, one of the things I'd like to hear you discuss about is, as these discussions have evolved about the interaction between anatomic pathology, our various histology sites across the Fairview system, and BioNet, my question is there a plan or have there been discussions for how to have interaction between those two laboratories? And let me expand on why I'm asking that question. So there could be studies, undoubtedly there will be studies, where the types of material, the types of cuts are going to be significantly different than simply getting, say, 10 4-micron unstained sections to do IHC on. Especially if you're talking about studies that are going to try and combine IHC with molecular studies. We're talking about different types of tissue, or different thicknesses of cuts, different ways of preparation, different slide types, can get relatively complicated. So is there a plan in place where blocks might be released to the BioNet Research IHC core where that could be accommodated a little bit easier or on a little bit more direct basis rather than the very, very busy clinical histology labs?
Excellent question. Thank you, Andy, for that. So if this is the case here, when the researcher fills this form, this is a pathology form. So if you are filling this form, all you can say, I want the block. You can mark block here. Where's block? Okay. And then you write down here, hand the block to BioNet for doing whatever. Okay. So your study pathologist will make sure that that block is only a representative. It's not like our Mrs. Block story. And we'll release it to BioNet, and BioNet can help you with whatever you want to do with it. Absolutely.
So that leads right to question number 2. So obviously, as your anatomic pathologists start doing more research, some of us will be requesting cases that we didn't sign out. Or in my case, all of the cases, which I don't sign out, since I'm an anatomic as a molecular pathologist. So for pathology specifically, or let's say Scott gets Paul Ray to work on some projects in prostate, that study pathologist, number one, is there a problem with them being the PI as well? And number 2, I'm assuming you had mentioned somewhere in there that you want the pathologist that had signed the case out to re-review it, but obviously that's going to go across. So
no, I didn't. I wasn't, and I apologize if I sounded like I'm implying that the pathologist who signed the case has to review it. I didn't say that. The study pathologist definitely can be different from the one who actually signed the pathologist. But going back to that thing that you mentioned about the conflict of interest, or are we going to run into trouble if the pathologist is conducting the study? And I must say, we are all professionals, and we try to follow the guidelines as much as possible. There is an oversight, and this is my role. I have worked in a previous place where even that process was very strictly regulated. I don't see the need for this here. Maybe 10 years down the road, if we get so big and we have like 50 pathologists, we need to do that. But today, within our family, I think we managed to stay in the clear.
Thank you for this talk, because having been, as you already know, a person that's trying to navigate this makes things just a little bit more clear. But as a person that has started navigating it, and researchers that may be at that point, do you think it's beneficial for us to get into Encore and at least put where we are, put ourselves into that system, rather than being having navigated this outside of this whole structure at this point in time? Is that a good idea?
We talked about that, yeah.
Okay.
We don't want to just tell everyone, sure, start putting your studies into Encore. It's a regulated process. Okay. Are you using Encore for any?
No.
Okay.
I've actually been working with Dr. Khalifa because I approached him originally.
Okay.
What we were trying to do. But I'm wondering, does pathology want to be able to, and do all the teams want to be able to bring all of us in to the system so the documents are there?
Eventually, yes. Eventually, everyone will be using Encore if they need any services. And, well, university-wide, any studies, they're going to want to be in Encore. right now there's a transition from tasks. I don't know if you're familiar with tasks into Encore, but it's very regimented, so we don't want to just tell everyone, sure, start. And you won't want to do that either. You'll want training.
To,
Can I just have you expand on that? So, sorry, as that transition of tasks into Encore, so basically for the next period of time, I'll let you answer what that period of time is. Those who have active tasks protocols should continue to use those.
To use tasks until directed otherwise. Yes.
So two things here. First of all, Encore is not ready yet because they still need to develop the question and they need to adjust the calendar development. Second is, as I mentioned, we try to make the process so user-friendly to allow you to enter into the post-award phase even without being on core. Just get the form and the new form and send it to pathology along with your IRB approval and set up the account, and then you get in the process from there.
And then my second question was, when you put in your CPRC application for that process now, of course that's changing now, and there's now a committee prior to the CPRC review for your IRB application. They do ask you if you want to put your patient accruals into Encore or REDCap. So as someone that's just dealing with tissues then, probably makes sense hearing what you said to go into to Encore.
Yeah. That's the hope. Yeah.
Okay.
So there are a lot of things in process. And I guess my question is, when do we know that they've been processed? Or how do we know that they've been processed? Where here's the final, let's go with this. And I know it's complicated and there are many moving parts, but are there going to be ways in which we can be notified?
Absolutely. There is going to be a mechanism where this is going to be advertised across the entire organization. I'm not sure yet what that mechanism is going to be, but probably through Fairview and through UMP at the same time. But when everything is ready and we move, we will need to send a very clear communication as to what the new world should look like.
This is an awful question, but any estimate of time?
Yeah, well, time is, we are very close, I would say, maybe a month, maybe.
Oh, wow, okay. Yeah. And just to restate the obvious, you need to bring a pathologist in early. You know, people who buy tissue microarrays, that has some value at some point. But I read a lot of grant proposals where they show you a tissue microarray and say they bought it, and here's what it is, and here's what we're thinking. It doesn't carry any weight because a pathologist hasn't weighed in on some of the subtleties of these tissues. And as you can see, it's extremely complicated. And to bring a pathologist in is only to your benefit. And a biostatistician, I would assume, so you know how many samples you want to have and what your question would be. So I would just emphasize all that's been said here. Utilize pathology. They're a valuable resource. I don't get any money for that. So. And one other, the issue of authorship, it's a fundamental problem, not just for pathologists. Because authorships are the most basic form of currency in this business. And the question is, what are you going to pay to have somebody do something on your study? You need to negotiate that first so people understand exactly what the rules are. And then you need to honor that agreement. So, I guess that's all I got.
Yeah, but we understand that in many of these research projects, the role of a pathologist is so small and it doesn't really rise to the level of academic acknowledgement. But in these cases, there will be a professional fee.
Absolutely. Yeah. And just to agree on it up front.
Yeah,
so
exactly.
That's the take home for me. Okay.
Anyone else?
Thank you all.
Thank you so much.